So gene networks are the networks of the genes. Before we go into the gene networks, let's see what the networks are. So network is a topology that connects its components based upon some relations with one another. Networks are implemented as graphs. Graphs are abstract representation of a set of data objects which are somehow linked with each other. And directed graphs have the orientations as compared to undirected or non-directed graphs, and they don't have orientations or directions. Here are the directed graphs. So we see the directions. We have, for example, this gene A to B, there is a direction. Then we have B to C, we have bidirectional thing. So it's two-way directed. And then from this B to C, we have again, there is a directionality. Undirected, or we can say bidirectional graphs, are where we don't see those directions. They may be like this, they may be where we have both the directions. So most of these gene correlation networks we see in some later slides, they are actually this type. Whereas the gene regulatory networks, the networks of genes are based upon their regulatory interactions with each other, and generally they are implemented as directed graphs. Two genes are connected if the expression of one gene modulates the effect of other gene by either activation or inhibition. So genes, they affect each other, they activate each other or inhibit other genes. So in that case, we can draw their gene networks. How can we model the gene networks? So we can have correlation or co-expression networks, we can have Bayesian networks, we can have use these differential equations, we can have Boolean networks, and we can have some what we call them as Gaussian models. In this course, we will only take these correlation networks. So for example, in our data set, we have gene 4 and 5. If we plot their expressions with one another, we can come up a line or fit a line on them. So that line is where we can see that the expression of one gene is increasing while the expression of the other gene is decreasing. So there is kind of a negative correlation between these two. So which one is affected which one? Either maybe this is going down, so that's why it's going up, or this is going up, so that's why it's going down. So in both cases we cannot decide. So there is a negative correlation between these two genes. So R is a negative number, fit in this line, and we fit in these regression line and come up these numbers where we do have these what we call them as slope and intercept. I'm not going into the detail of this regression model. So here we are just looking into this correlation coefficient. So they do have strong negative correlations. Correlation may be a negative or a positive. So this kind of situation where they have a negative correlation, the value is close to -1. So correlation ranges from -1 to +1, 0 in between, that means no correlation. Here we look into their expression profiles or expression patterns. So we have G4, it's stable, and then it's going all of a sudden it's going down, but at the same time the expression of G5 is going up. So in this way we can say that they have some opposite effect on each other's expression. We take another example. So we have G4 and G5. So here we have correlation which is equal to 0.998. We come up with the correlation value that is equal to 0.998, which is approximately equal to 1. So both of them are increasing in expression. So there is a strong positive correlation between these two. In fact, G1 and G3. Here we see another pair of genes, G1 and G3. We see that both of them, they are positively correlated with one another. The correlation value is 0.998. Both of them are increasing. As far as A is increasing, B is also increasing. So we fit a line which is like this. If you look into their individual profiles, we see that both of them, they are following the similar pattern. So they are increasing at the same rate. Though there is the difference in their magnitude of expression, Pearson correlation does not take into account, but there are some other correlation methods like Spearman, etc. They also take this difference in magnitude of expression into account. So we can have those gene-wise, gene-to-gene pairwise correlations. We can come up with a correlation matrix, and we can use this matrix to draw their gene networks. This matrix can also be represented in the shape of these heat maps, where for example red ones are where we have higher correlations are positive correlations, strong positive, sharp red, and then we have strong negative, it's sharp blue, and then there are shades in between them. So this representation is called as the heat map representation. For instance, if you have these correlations, so we have G1 which is connected to 4 other genes, G2, G3, G4 and G5. So we can use these correlations and draw those edges which are actually presenting those correlations between them. Green edges if there is a positive correlation, and thickness of edge may be correlated with the strength or the magnitude of that correlation, whereas this negative correlation is represented by this red color edge. So in this way we can put them together in the shape of the network where we have those nodes, the genes, and the edges or the connection between them representing their correlation values. So gene networks are abstract representations of relationships of the genes based upon their gene-to-gene correlations, and where in these networks the genes are your nodes, whereas those correlations are your edges.